Good morning, everyone. Very welcome to another session of the seminar series at the Fraser Institute. First of all, I would like to acknowledge the country. The University of Queensland acknowledges their traditional owners and their custodianship of the lands on which we meet. We pay our respect to their ancestors and their descendants who continue cultural and spiritual connections to the country. We recognize their valuable contributions to Australian and global society. And we are very proud of the diversity we do have in general in UQ and specifically at the Fraser Institute. Please scan the barcode to register yourself as audience in the seminar series. Okay, I'm very happy to introduce Professor Jason Roberts, who's the Deputy Director of the Fraser Institute and Interim Director of Health and Infectious Disease Institute. Academically, he has an Australian NHMRC Leadership Fellow and also a Fellow of the Australian Academy for Health and Medical Sciences. Clinically, he is a critical care pharmacist at RBWH. Jason has a significant academic and clinical interest in optimization of antimicrobial dosing regimes and is globally ranked first for publications for keywords pharmacokinetics and drug monitoring. So very happy, and Jason is going to introduce other speakers for us. Thank you.
Thanks, Behzad. It's very nice to be presenting at this seminar series and to see so many people here in the room. So I work for a group called Antimicrobial Optimization, which is one of the groups within the Fraser Institute. And of course, we're based here at Hurston, and we're based here because we have a number of people that are also clinicians that work at the Royal Brisbane Women's Hospital. And so that's a very good thing for us because it gives us access to people that are able to describe to us the clinical challenges that they come across. And where it's relevant to what our skills and capabilities are, it allows us to work with them to help design solutions to those problems. And most commonly, that's related to how to best use drug therapies. Of course, there's a bit of a perception that once we've made a diagnosis, that we give a drug therapy and that drug is always going to work as well as what is intended. But that's not always the case because the way that drugs behave in patients, which means the different concentrations which may manifest, can be quite different depending on the patient population and the dose of drug that's actually selected. What I learned very early on in my clinical practice is that there was a fantastic heterogeneity in the dosing of antimicrobials that was used in practice. So I'd see very similar cases, completely different decisions being made by the prescribers, and it became very quickly apparent to me in discussing it with them that they were guessing. They didn't really know because clinical trials and clinical studies hadn't been done in those patients for them to be able to have some evidence base to rely upon, and so they had to use their judgment, which was based on previous experiences that they had. And so I thought, well, this is a great opportunity for a research program to help provide evidence to address these challenges. And so that's something which we've very much focused on. And so with this, we've been able to establish a very nice group of collaborators internationally who have been able to help us answer many questions, which are problems that we see locally here, or alternatively, are issues that they see locally in their hospitals, whereby we're able to support them finding solutions, or alternatively are just international challenges which really do need a coordinated partnership approach to find solutions to those challenges. And so with that, we were able to establish a Centre of Research Excellence, which was initially formed back in 2015 and was renewed in 2021 as well. And so we've been fortunate to have a Centre of Research Excellence from NHMRC renewed, and that provided us with some really strong machinery and infrastructure to be able to support a lot of these collaborations. And so, I'm very proud of the amount of collaborations that we've been able to enjoy in the past, and a lot of it's been because of that CRE. And I think that we've done a great job in terms of what the aims of that funding scheme are to make sure that we deliver. Anyway, in terms of what our program of research are, we've tried to make it as end to end as possible. So from the most basic pharmacological studies all the way through to clinical trials and post clinical registration validation studies. We've tried to capture all of the different steps that are required to understand what pharmacokinetics a drug has. So pharmacokinetics is essentially the study of what concentration of drug is measured in a patient based on a particular dose that they've received. And so we have what we call in vitro PK/PD models, which is looking at in a benchtop scenario, what is the effect of differing drug exposures on the ability to kill or inhibit the growth of bacteria and also to suppress emergence of antimicrobial resistance. We have things like ex vivo models, which help us understand what the effect is of clinical practice modalities such as dialysis or extracorporeal membrane oxygenation or nebulization of drugs to see what kind of dosing we should be using if we should choose to if the patient requires renal replacement therapy or dialysis, if they require ECMO, or if we want to enhance lung concentrations, how do we aerosolize that drug most effectively? We do studies with volunteers and in clinical pharmacokinetic studies, and certainly clinical pharmacokinetic studies is where we've done most of our work, and we would have done a lot more than any other group in the world in that, perhaps at least 50% more than any other group in the world. So that's where we've done a lot of our work. But our clinical pharmacokinetic studies are very much underpinned by a very strong capability in pharmacometrics, which is essentially the data analysis component of clinical pharmacokinetics. And this is where what Jin will be talking to when she talks about ProSNAP, and the clinical pharmacokinetic studies will be discussed by both Patty and Hafiz a little bit later. There are also clinical pharmacodynamic studies, which essentially are studies which look at measuring concentrations of drugs in patients and comparing those concentrations with outcome to try to understand, well, what are the best concentrations that we should be aiming for in patients to then allow us to design studies which can see if we can further improve the effectiveness of those drugs by, in a prospective trial, by seeing the effect of achieving those concentrations which have been defined in those clinical pharmacodynamic studies. And then there's some clinical trials as well, which I think Hafiz is talking about. Are you talking about Bling3? I've forgotten. Yep, great. Now, as you can see from all of this, like, I am pretty hopeless, but I have an amazing team, and that's why they're going to be doing the majority of the speaking, because it's through a really functional team of excellent people that we've been able to be very effective in our area of PK/PD research. I'll also just highlight that we have an education program which is underpinned by funding from that Centre of Research Excellence as well, which has delivered a really extensive program of work, over 50 seminars, inclusive of nearly 200 lectures, which we've actually made available on YouTube. And so we have our own YouTube channel. And we also run workshops for performing pharmacokinetic modelling as well. And they've been very successful, and we've been invited overseas to provide those as well. And so that's great for the university, great for us, and also great for developing broader capability for these techniques around the world. So this is our group, and I just thought I'd show you this just so that you can see the breadth of work that we have across here. I'm really proud about a lot of our groups which transcend consumer engagement. And I've covered a lot of these other areas too. I will highlight our bioanalysis lab, which is really state-of-the-art, perhaps with non-state-of-the-art equipment that we own, but we have access, of course, to the mass spec facility, and we are trying to get access to better mass spec equipment, which we can have ultimate control over. But Helen and Dushan and Jenny and Tin do an amazing job with huge volumes of samples that come from all over the world to be able to deliver a really strong bioanalytical capability. And we're very proud of all our PhD students, all who progress very well and do fantastic work.